Okay, welcome back, everyone. Good afternoon, and welcome to this post-lunch and final session of this meeting. So we'll have two talks, and the first one will be given by Kedar Damle from TIFR Bombay, and he's going to talk us about chaotic percolation in interesting settings.
Thank you. So it's very nice to be back here, of course. So thanks to everybody who organized this. The title is a bit of a mouthful, but I couldn't think of any way of compressing all the information into fewer words. So I'm going to talk about a percolation phenomenon I'll tell you what these are. But the reason I'm sort of daring to talk about it here in a meeting on fractionalized quantum matter is that I'm going to claim that there are some implications for spin liquids and for topological superconductors, which are in principle measurable. So that is the real punchline as far as this audience is concerned. If this was a StatMech meeting, I would have focused entirely on this part. So, and this is all work done over a number of years with a bunch of students, starting with Sambuddha Sanyal way back almost 10 years and then progressing. And the most recent work is with Zahid Ansari, Ritesh Bhola, and Shauvik Kundu, who's somewhere in the audience. So let me start. So just a few generalities. So disorder, quench disorder, by which I mean some, missing atoms in the crystal, some add atoms, some crystal imperfections, which don't move on the time scale of the experiment. These are quite common in condensed matter systems, although you'd like to get rid of them to look at pristine materials, it's not always possible. And the effects of such quench disorder is quite varied. So if it's very weak, it can sometimes be quite irrelevant for the low energy properties, but not always. There are situations in which very weak disorder also can change the behavior. For example, at some phase transitions. If the disorder is very strong, you can actually get new phases of matter. For example, spin glasses, right? Much of glassy behavior in spin systems comes from disorder, strong disorder, in which the sign of the exchange constants also are random. And a more interesting aspect of disorder from the point of view of probing the pristine sort of underlying state, the quantum many-body state, is that sometimes by having a little bit of disorder and knowing exactly what it's doing, you can use it as a probe to probe the correlations of the underlying pure system. And there's many examples of this in frustrated magnets that many of you are familiar with. And so underlying all this is sort of a central dogma we more or less absorb when we are students. In its strong version, it would say that if you're in the large size limit, then physical properties that you measure in a macroscopic system are going to be self-averaging. So their sample-to-sample fluctuations are going to be small relative to the mean. In other words, the average over 100 samples is going to be more or less the same as the typical value in any one. So average equal to typical is another way of saying this. And there are violations of this. For example, it's now known that if you have a random exchange quantum spin chain, like a spin half chain with random antiferromagnetic exchange, but which always remains antiferromagnetic, it's been known that this kind of system at very low energies flows to what theorists would call an infinite disorder fixed point, and now you get broad distributions. So the average value of some physical observable, like a susceptibility, is not necessarily the same thing as the typical value. So if you look at the distribution of the physical observable, the distribution over the ensemble of samples, there'll be a typical value, which is where the distribution peaks, but the average will be controlled by some long tail. This is known to happen, right? So the strong version has violations, we know them. But there's a weaker version of this sort of central dogma, which is the statement that at a minimum, two samples prepared using the same experimental protocol with good control over what you're doing should be in the same phase of matter. That much at least we usually grant ourselves. So are there violations of this? Well, maybe in some infinite range spin glass models, there could be. And this is not a subject I understand very well. It's also been a subject of some controversy. So let me not go there. But this is something we more or less take for granted. So my basic message to you here is that there's a perfectly natural system one can look at where in the weak disorder limit, you don't even need strong disorder, you have violations of not just this strong version I was talking about, but also of this weak version. And so basically you make two samples in an identical way. One of them will have, will be in a certain well-defined sense, be in a different phase of matter from the other. This can happen, is the claim. That's one claim. And we think that the underlying reason is that there are kinematic constraints in the dynamics of this system, which induce some sort of long-range correlations that are responsible. So that's just sort of post facto rationalization. We don't really have a detailed understanding, but this seems one thing that's at play. So, like I was saying, where's the fractionalized quantum matter in this story? Well, I'm going to claim that what I just said in the previous slide implies similar effects due to weak vacancy disorder in triangular lattice, short range resonating valence bond spin liquids. If you have a material that is in that phase of matter on the triangular lattice, a spin half material, there is a claim that it will show some such effects. There is also the second kind of example of this is that if you have a topological P plus IP superconductor, where we know that every vortex carries with it a Majorana mode in its core, then this multi-mode system of Majorana fermions will be in two different phases, in two different samples, more or less identically prepared, if there's vacancy disorder in the pinned vortex lattice. So by vacancies here now, I now mean missing vortices. And the same thing is expected if you have a triangular lattice Majorana spin liquids. These are the sort of statements. And a bit more concretely, in the short range RBB case, at a minimum, you're going to have strong violations of thermodynamic self-averaging in the low temperature susceptibility. And quite likely, although this we don't know for sure, two kinds of samples, which you later see I call R-type or P-type. One kind of sample will have spin glass order, the other will not. And you prepared them identically. You don't have control over which one's which. Right? That's the claim. In the pin vortex lattice state, the claim is if you measure the thermal conductivity at low temperature, again, there'll be some violations of thermodynamic self-averaging, and likewise here. Okay, so that's the connection to this meeting. So what is the setting? What am I actually studying? What I'm studying is maximum density dimer packings of diluted lattices. So that's a lot of adjectives. So what's a dimer? It's a hard rod. You put it down on links of the lattice with the rule that no two dimers touch at a vertex, right? That's the hard constraint, the hardcore constraint. What's maximum density? Well, you try to put as many down as possible, subject to the hardcore constraint, right? So here's a small fragment of a diluted triangular lattice. All these light gray dotted lines, they don't exist because the corresponding vertices of the triangular lattice have just got knocked off. Those are the vacancies. It's quenched. So this pattern is fixed, the underlying lattice, right? That's the quenched disorder. So here so there's the only surviving sites are the dark blobs, and the links between them are the dark lines, right? So it's just a small example I constructed by hand to illustrate a few points. So you try putting these hard rods down now, and I've done it in one way, and I claim this is the maximum density I can reach. So we'll see that in a minute, right? So sorry? Right, so we'll play with this. Actually, it turns out in this particular example, you can't really get away with pure monomers, right? So what are the takeaways from this? Well, first thing, notice that this is by no means unique. Like over here, in this little part of the surviving sample, I could just do it in a different way and I would get one more, for example, I can keep this the way it is and just rotate these two so that they become vertical. So there's some local rearrangements possible. That's one, right? But at the same time, like Subir was asking and I was saying, you don't seem to be able to make enough local rearrangements, although I don't do this exhaustively for you, you can check it, to get rid of the monomers. Okay. So from this picture, I would conclude, if I have a pure lattice with no disorder, then it's easy to find a fully packed configuration with no monomers. And that's because, you take some local motif, find some way of doing it perfectly there, and then by the translation symmetry of the lattice, just repeat that everywhere, right? And if that local motif has more than one way of doing it, you also realize then that there'll be a non-zero entropy density of such fully packed diamond covers. So in the pure case, there's no problem, right? Of course, if it's a bipartite lattice, so that there's two kinds of sites that are always next to each other, you will need their numbers to be equal because a dimer always connects one A to one B. But on the triangular lattice, there's no such thing, right? But with weak vacancy disorder or bond dilution or any kind of dirt, right? This example suggests, and it turns out to be true, that you'll typically have a non-zero density of monomers, just required, right? By the combination of the hardcore constraint and the maximum density constraint. And you'll have, again, a non-zero entropy density of such maximum density dimer packings. So that's one takeaway, okay? So what are the constraints on such maximum density dimer packings? So again, let's go back to this picture. You see, if I want to go from this choice I made by just playing around to something else, which is also maximum density, I can only do two things. I can either hop a monomer like this. So, this dimer rotates here, this one rotates there. And in effect, what happens is that the monomer has gone there, right? I can do that. So that's a kind of a monomer hopping along an alternating path. What's an alternating path? Well, alternate links are covered by a dimer, not covered by a dimer, right? So you can hop a monomer along an alternating path. You can also have alternating loops around which you can flip the state. So here, this dimer goes here, this one goes here, this one goes here, right? So this is what's called ring exchange. So you can have ring exchange on some alternating loops. You can have monomer hopping on some alternating paths, right? These are the two ways I can mess around with this picture and retain the maximum density and the hardcore constraints. Now notice that each of these possibilities requires one thing, that there must be some maximum density dimer packing in which all the links that are part of the path or the loop have the possibility of being occupied by a dimer. That's a requirement, otherwise, that's what's happening here, right? Things that were not occupied are becoming occupied. So these are sort of the two takeaways from this picture again. So more conclusions. There's constrained kinematics. And the constraint on the processes that are allowed is that each link along those paths or loops must have the possibility of being occupied by a dimer in at least one such dimer packing, right? And one more thing you can notice here, and this goes back to Subhi's question, let's try concretely to see where I can make this monomer go, right? I can make it come here, clearly, or even there, or there, and so on, right? So it can more or less move here, but I don't seem to have a way of making it go there, right? Similarly, this monomer, I don't seem to be able to get it to go out. So somehow this region, I seem to be forced to have exactly two monomers, no matter what I do. This region, I seem to be forced to have one, and this seems to be always perfectly matched, right? So these are just, observations. So the idea is that, yeah.
Starting from that configuration, you can't even change the model.
I'm just saying you make any maximum density dimer packing of this fragment. This blue region is always going to have two monomers.
Is it clear that given this lattice?
Given this lattice, the lattice is quenched. That's quenched disorder.
So given this, the number of monomers is fixed by the maximum.
Yeah, but more locally, certain regions must have certain numbers, not just the global number. Yeah. Well, not in this example, obviously, because if I had weak disorder, I would need to construct a humongously large picture with just a few vacancies, right? Here I've put a lot of vacancies to show you what's going on. Yeah. So monomers seem to be confined to well-defined regions, and other regions are fully packed in any maximum density dimer packing. So let's go further. What is it that tells me that I can't go here, right? I can't send a monomer out. If you think about it, it's the fact that you can't really have, I think it's more obvious with this picture. You can't really have a maximum density dimer packing which has a dimer on this link. Because think about it, if you did that, well, you would end up having one monomer here anyway, and you would introduce a third monomer on this one. So there are certain links of the lattice that simply cannot be occupied by a dimer in any maximum density dimer packing, right? That's the third observation. So these boundaries of these monomer carrying regions, which I call R type, and the perfectly matched regions, which I call P type, the fully packed regions, they're actually defined by these forbidden links that can be never occupied. Okay, so these are the conclusions just from these pictures. And this is all true. I mean, I wouldn't expect you to believe it based on one small picture, but one can prove this, right? So there's a formal justification for everything I said. It uses a prescription or a theory that goes back many, many decades to Galai and Edmonds, computer scientists. Edmonds actually being the man who first defined this notion of P versus NP, polynomial time algorithms. And the algorithm he came up with for this problem was sort of the first one that was said to be polynomial explicitly. So anyway, the prescription, how am I doing on time? Left. Okay. So the what they say is, pick your favorite maximum density dimer packing, and you just now form these alternating paths that go outwards from every monomer of that matching, right? Of that dimer packing. And you just explore that forest of paths. And you label vertices of the graph in the following, where did I go? In the following way. If you can go along one of these paths starting from a monomer and reach a vertex with an even number of steps, you will call that even, E. If you can reach it in an odd number of steps, but not an even number of steps, you will call it O. And if you can't reach a vertex, you will call it U, unreachable. That's what they say. This is the prescription. You do this. Then the theory says, and it's not very hard actually, there's a lot of combinatorics, is that this labeling of vertices of the graph of the lattice is completely independent of the choice of maximum density dimer packing you made. So you could have drawn something else which was maximum density and done this. You would have got the same E, O, and U labels for every vertex. That's guaranteed. That's the theorem actually. The Galai-Edmonds theorem just says that. Okay. And it also comes with some structural guarantees about the disordered lines. So for example, it guarantees that there can be no links of the lattice between E and U vertices. And also that when you delete E and O vertices, it organizes all the E vertices into sort of connected components that don't talk to each other, which all have an odd number of vertices inside. And some other some things. And all the monomers in any maximum matching always live on E. And each of these connected components are called blossoms by them. So this, I couldn't resist putting a picture here. This paper is called Paths, Trees, and Flowers. So you go along this forest of alternating paths, starting with a monomer. If you can reach some vertex with an even number of steps, you call it E. Yeah. There's a slight subtlety. Many vertices that are even, most all vertices that are even, can also be reached by odd. So odd is you can reach it by odd number of steps, but not an even. So yeah, so that's a further thing I didn't say, but I wrote down, I think. So all the monomers live on these connected components of E that you get by deleting EO links. And no blossom has more than one monomer. I didn't say everything has one. It can't have more than one.
Because you define vertices, you classify vertices based on some path from one.
No, but the question is, do you always need a monomer on every blossom? The answer is no. This is a precise statement. All monomers live on blossoms, and no blossom contains more than one. It can be 0 or 1. Right? These are all things you can check with a little bit of work. I mean, this proof is not hard. The idea is very wonderful, but the proof is not hard. So we use this and say, look, if I delete the OO links, and the OU links, these are the forbidden links I was talking about in that picture. So the minute I delete them, the graph or the lattice breaks up into components. All the components made up of U are the p-type. They're always perfectly matched. And all the components that are made up of O and E, right, are my R-type regions. So the forbidden links, the main observation we make, and this is the only thing we need to do actually, and it's quite remarkable, is to realize that OO and OU links in their theory are precisely these forbidden links. Okay, so this defines for me the regions of the graph, and it also tells me how many monomers live in each graph, in principle, right? That's the background. So why do I care? I mean, did I just start trying to find some problem to, get confused about? Well, not quite. This has implications, right? We got to it because of the physics. So if you start thinking about quantum monomer dimer models, for example, this is the most theoretical of the motivations, so bear with me. You realize that none of the monomer hopping or ring exchange processes people will write down would cross any of these boundaries, right? So the dynamics is factorized. So any Hamiltonian you write down, which has any kind of dimer interactions around flippable loops, any kind of monomer hopping, any distance, but short-range monomer interactions, not beyond third Weber or something, right? All the eigenstates of any such quantum dimer monomer model are going to be completely factorized. The problem on each R-type region is independent of other R-type regions. The problem on every P-type region is independent of every P-type, right? So this implies if all the regions are small, you're going to get area law entanglement everywhere in the many-body spectrum. This is not MBL, but this does happen. But that's sort of the most theoretical of the motivations. There's more practical motivations. Sorry? Has to be for this to work? Well, I don't want to get into that now. That's slightly technical, right? Okay. Basically, you have to ask yourself, when can monomers in two different R-type regions interact? That sets the range. So you have to do a little more work, right? I think it's not beyond second or third. But this is a vast class of Hamiltonians. But anyway, that's just pure theory. Let me say something that's perhaps more interesting to many people. If you had a short-range resonating valence bond spin liquid, there's a common caricature of it that comes from Roxer and Kivelson's work, who wrote down a quantum dimer model. The idea was that you don't work with one pattern of singlets between spin halves, but you say the system is sort of resonating between all possible singlets. That's the resonating valence bond. And short range simply means, at least in this cartoon, they're nearest neighbors, but you don't have to take that literally, right? As long as it's short range, I think what I say works. Within this framework, at least if I take this cartoon seriously, every monomer I was talking about is going to be associated with an emergent local moment that sort of is there at low energies in the system. So well below the gap of this spin liquid, right? And it's going to be confined to these R-type regions. And so these emergent local moments are a multi-vacancy effect. They're not some trivial thing where I knock a vertex off and so the singlet that was being made can't be made. It's not like that. They're a multi-vacancy effect. I mean, you saw that already. That one R-type region had many vacancies around, right? And the dominant short-range interactions between these local moments are going to be confined. Mostly, they're going to act within each R-type region. That's just because there's this gap in the system. And so, therefore, the geometry of these regions is really expected to determine what happens at low energies, right? Below, much below the spin gap of the pure, spin gap or singlet gap of the pure system. And you should contrast this with a more familiar example of a lattice symmetry broken VBS state, where, of course, it's much more trivial. If you just knock a vertex off, there's a non-magnetic impurity there, there's going to be a local moment just bound to that vacancy. That's trivial, right? This we know from a long time. So why is this distinction really happening? Because at least the heuristic picture is that in the VBS state, there is a favored ordered pattern of valence bonds the system likes to make to minimize the energy. So although there are full packings available. That means you can make more singlets. You don't need to leave some things unpaired. If you exploit that possibility, here's a full packing, right? Full density. If I exploit, so these are the vacancies, and I've exploited the fact that there are other possible ways of making singlets, I create a domain wall. So this is going to cost energy linear in the size of the domain wall when there's long-range spatial order. So the system doesn't do it. Every time you remove a magnetic ion, it creates a local moment. But in the RVB case, there is no favored pattern. As long as you can do something and there's an entropy density of things you can do, that's a good wave function, variation. That's the heuristic idea here. But this also sort of follows more formally from formal arguments relying on large N expansions. Subir is the one who initiated those ways of thinking. So, and also you can check that this works via explicit quantum Monte Carlo on model Hamiltonians that have valence bond or spin liquid states with some enhanced symmetry. Okay, so I won't go there. So conclusion, you have this local moment instability of RVB and VBS states. It's kind of trivial in the VBS case. Every vacancy has a local moment. These local moments interact. But in the RVB case, you're only going to get this kind of instability if the density of monomers in the maximum density packings of this graph, of this lattice, is non-zero, right? In the VBS case, you're going to get it anyway. Each vacancy is going to give it. This maximum density or full packing doesn't matter. And then these local moments are going to interact with some effective interaction, and the ultimate fate of the system at lower temperatures is determined by what happens to this network, right? That's the idea. There's one more sort of problem that brought us to looking at these maximum density dimer packings, and that's simply the Majorana Hamiltonian, free Majorana fermions interacting bilinearly with each other, right? This is the analog of the free fermion problem if you have Majoranas. This is some purely real antisymmetric matrix. That's why there's an I there. And these are the Majoranas. Now, this is known to be the good description of topologically protected Majorana zero modes. For example, in this vortex lattice of P plus IP superconductors, each vortex has a Majorana mode, they mix. This is the mixing Hamiltonian, right? Likewise, for triangular lattice Majorana spin liquids, Subir and I think Rudra Biswas have a paper on that. So, sorry, I didn't say what the point there is. Now, if you look at this, you can ask, can I make linear combinations of these that have eigenenergy zero? So they're fixed to zero energy, which means they are themselves Majorana modes. They're collective Majorana modes. And the answer is each R-type region that hosts, let's say, I monomers. Remember, the number of monomers in an R-type region is fixed by the quench disorder, right? It's whatever lattice you want to. It's general, right? At this level is general. The A's define my lattice. Yeah. So you give me the mixing matrix and I draw the graph, right? That's all. So for example, if you had you had a vortex lattice, and you just said the dominant mixing is nearest neighbor, then this would be a triangular lattice. For your triangular Majorana spin liquid, the mean field theory at least is just this on the triangular lattice in the pure case. You don't have that to begin with, you can put that in. Right now, it's not there, right? If you put that in, that's a different graph. And I'll do everything I'm saying on that graph because I'm anyway going to talk about dilution. So the parent graph is not so crucial, right? This is sort of the general framework. Statement is you take whatever is the graph you get from this, and there's going to be some R-type regions with some number of monomers in each. And those are exactly the number of topologically protected collective Majorana modes in each of those regions. The wave functions are localized there. That you can prove. That's not hard. So these are the connections to what this audience is interested in. And with that, let me just say, this motivated sort of a large-scale computational study. Yeah. So locally, the number equals number, but I have you can produce a prescription that you can code on the computer to work out the wave functions as well. So you'll have these many linearly independent wave functions living in those region. This doesn't mean much, right? If you have a huge degeneracy, you can take any linear combination and that's also a good wave function. So what I'm really saying is I can make a maximally localized basis of the null space of this matrix, such that each basis vector lives entirely within one R-type region. That's what I'm saying, right? So any basis independent property, this is a good basis to calculate. That's the idea. So we then just did numerics and looked at this random geometry of these R-type and P-type regions, specifically for the diluted triangular lattice. Do I have 5 minutes? Perfect. So let me now tell you what we see. So I'm going to talk mainly about low dilution. I should say one thing. We find that the typical regions at low dilution are huge. So we want to think in terms of percolation, right? And what's percolation? Well, basically it's a sharp threshold for answering this yes-no question. Is there end-to-end connectivity in the sample? Can I walk within one region entirely and go from one end of the sample to the other in the large size limit? And in the large size limit, that's a sharp yes-no question, and it has a threshold behavior for the answer. Answer switches from no to yes, and that's a phase transition, that's percolation. So now let me tell you what we see. So first of all, this is that probability of being able to cross. And you can see, so I go from sizes that are 10,000 by 10,000 all the way to sizes that are 22,000 by 22,000. And this is the density of vacancies. At some fairly high density, but below the percolation threshold of the underlying lattice, there's a phase transition of these R and P-type regions, so that below this critical point, at lower density of vacancies, you can go from one end of the sample to the other with probability 1 in the thermodynamic limit, but above you cannot, walking entirely within one of these regions, right? Now, we are interested in the low density phase because I'm not interested in 48% dilution. I'm thinking of 5% dilution, weak disorder. So what happens when the disorder is weak? What we find is that if you just look at the mass density of the part of the lattice that lives inside R-type regions, that looks very noisy but has no vacancy concentration dependence. I didn't show you the graph that goes further, but here's the inset. So all the way down to 20% is just a flat line, but very noisy. And this is not because we were doing crappy numerics. We took actually huge ensembles, but it's still very noisy. And so we were wondering what this is, and we realized it's because it's bimodal. If you look at the mass density, half the samples roughly seem to have a huge mass density. Most of the lattice is R-type, and half have a tiny density. So there's this, already you see that there's two different things happening in two samples, you prepared them identically, right? But the density of monomers, the overall density of monomers is just smooth, doesn't show anything. That's the first observation. This is all in the percolated phase at very weak disorder, right? So now we look more closely. We call all samples that belong to the right peak, that means most of the sample is R-type sample. The other samples we call P-type. So now I break up my ensemble of samples into two types, right? So if you look at an R-type sample, it's got, and then I look at the largest R-type region in an R-type sample. It seems to cover most of the lattice. It's huge, it's macroscopic, and it has no size dependence in terms of density variance. So the size grows with L square. But in a P-type sample, it shrinks, the density shrinks to 0, so it's some order 1 region, doesn't grow with size. So that's a huge difference, macroscopic difference between the two kinds of samples, right? Similarly, you can look at the largest P-type region, and it's got exactly the opposite behavior. That's here. And you can check that this probability to go from one end to the other becomes 1 at low dilution. Why is it 1? Because in R-type samples, there's always one R-type region that spans the lattice. And in P-type samples, there's always one P-type region that spans the lattice. But it's completely different, right? Now, the really interesting thing for us was, this is all already mind-boggling, but the fraction of R-type samples relative to P-type is just flat throughout, and it's half within errors. 50-50. And so there was some worry that we are doing something stupid. So the one obvious property of the lattice as a whole, which is 50-50, is the parity of the number of vertices in the largest connected component of the lattice itself, right? You've diluted it. There's going to be one huge component that spans the lattice, and then smaller blobs. That's percolation theory. So you take that huge component, you count the vertices in it. That can be even or odd, and that's going to be 50-50. That's the only 50-50 variable we can think of. So we asked, is this 50-50 because of that? Right? And so here we have split the full ensemble of samples into an even ensemble and an odd ensemble. So the largest geometric component is even here, odd there. And it makes no difference. Whichever ensemble you look in, this FR is 50%. So this does not come from sort of the obvious explanation. There's something deeper here. I don't know what it is. And this is how the picture looks. This is the largest R-type and second largest R-type of a R-type sample and the converse. So you see it's just completely different, the two samples. Color coding is consistent. So this is a violation of even the weak form of this central dogma, right? Because the monomers are delocalized in half the samples, localized in the other half. And all samples are identically prepared. And so can we quantify this? Well, we try to set the vacancies into motion. A very small fraction of vacancies move at every time step. Like 5 into 10 to the -5 is the fraction of vacancies that move to a nearest neighbor, exchange positions with a surviving site. Very small number is moved, purposely very small number is moved, because we don't want the geometry of the lattice to change much in each time step. And then we ask what happens to the large scale geometry of these regions. And so this is proof that the geometry of the lattice doesn't change much. You see the fraction of vertices in the largest connected component of the lattice is just not doing anything. You can see what this scale is. It's moving in the 5th decimal place. Similarly for the monomer number, the total monomer. But the large scale geometry of these monomer carrying or fully packed regions just responds chaotically, you know. So what I'm showing here, sigma is a property of the sample. Is the sample R-type or is it P-type? Does it have percolating R-type region, percolating P? That's sigma plus minus 1, right? And gamma is this other variable that is whether or not the largest geometric component is odd or even. I want to check again that this doesn't predict that. This is the histogram of how many time steps it takes before the thing flips. So you can see after a few time steps of this dynamics, sigma goes from 1 to -1. R-type becomes P-type and vice versa. And similarly for this. The important thing is their product, which is what's controlling the cross correlation, also keeps flipping. There's no cross correlations. You can see that here. All cross correlations are 0. Right? So this is at least an operational definition of chaos. You have one macroscopic variable that's changing. In principle, there's a deterministic relationship between that and what's happening, but we can't predict it. And as I've already said, there are some observable predictions. And with that, well, there's some other stuff which I don't need to go into. So let me stop here. Thank you.
The interest of time, we have time for maybe one pressing issue. Okay. I'll just ask one. So maybe I missed it, but when you're labeling these E and O vertices, they were with reference to a particular monomer.
Yeah.
So have many monomers in.
Sure. So one minute. You're talking about this, right? Yeah.
So this is with respect to one particular monomer.
Yeah, so first thing to prove, which is very easy, that if you are E with respect to one monomer, you're going to be E with respect to anything else.
This is because of the bipartite nature of the.
No. This has not got anything to do with bipartiteness. Bipartiteness would make a clean distinction between E and O. The only thing bipartiteness would add to this story is that there can be no vertex that is E and O, both. Remember, if it's E and O, I call it E. This is like,
I see.
Right? If you can reach it both ways, you call it even.
Okay.
But on a bipartite lattice, you'll never reach it both ways. That's the only distinction. Otherwise, no. Okay?
All right.
All right.
Let's thank Kedar once more.